And our last speaker for this session is Michela Locci from the University of Pennsylvania. She received her PhD in Italy and trained as a postdoc with Shane Crotty. Their research aims to understand B-cell, TFH cell interactions, staying on the topic, and she will share her data on the immune response to mRNA vaccines.
All right. Hello, everybody. So first of all, I would like to thank the organizer for having me here. It's a great symposium and I'm enjoying it very much. So today I would like to share with you this, the work that my lab has done over the past few years, trying to understand the immunological mechanism of mRNA vaccines with a specific focus on germinal centers, which we already heard a little bit from the previous talk. And so in a nutshell, these germinal center reactions that take place in the vaccine-draining lymph node after vaccination are a very sophisticated reaction during which B cells undergo affinity maturation and they're selected ultimately to differentiate into long-lived plasma cells that secrete antibodies and memory B cells. And this process is tightly regulated by these specialized B cell helpers that are called T follicular helper cells, or TFH, which are initially primed in the T cell zone by dendritic cells. They can be found at the TB border as an early TFH precursor, and then ultimately as a mature B cell helper in the follicle. And so during vaccination, it's very important to induce this type of reaction because they're generally associated with high quality B cell responses. And my lab spent quite a lot of time during the past few years trying to understand how mRNA vaccines induce germinal center responses. And we actually started to work on the topic back in 2018 when I first opened my lab. And then thanks to our collaboration with Norbert Pardy. And then during the pandemic, like everybody else, we pivoted to SARS-CoV-2. And we were the first one to show that SARS-CoV-2 encoding mRNA vaccines in mice are really potent inducer of germinal centers that importantly correlate with the production of neutralizing antibodies. And then a few years later, we show, similar to data from Ali El-Lebedi's group, that the same is true in people. So humans with an intact immune system have in their vaccine-draining lymph nodes, which we probe by fine needle aspirates, these beautiful germinal center responses that are tightly correlated with neutralizing antibodies. And so it's clear that it's very important to induce germinal center in vaccination, in mRNA vaccination. And the question for us was, how is this process regulated? What is the mechanism by which these vaccines are so good at driving germinal centers? And I guess the first step toward gaining a mechanistic understanding was done a few years back in collaboration with Norbert when we asked the simple question of what is the adjuvant of mRNA vaccines? Because these vaccines are generally referred to as an adjuvanted vaccine. And so they don't need the addition of an adjuvant to be immunostimulatory. And so in this study, we show that really it's the lipid nanoparticles that work as an adjuvant, because if you take lipid nanoparticles and you mix them with a protein antigen, these protein plus MTLMPs drive germinal center responses that are as potent as the one driven by the mRNA vaccines. And they're also, MTLMPs as an adjuvant are also very good at driving the generation of memory B cells and plasma cells. And mechanistically, at the time we published this paper, we show that these depends, at least in part, from the capacity of the lipid nanoparticle to drive the production of IL-6, which is a potent pro-TFH-inducing cytokine. But so we published this in 2021, and we were left with many outstanding questions. And in particular, we wanted to know whether the mRNA vaccine component also is immunostimulatory, and if so, how does it shape the magnitude and quality of germinal center and germinal center derived responses. And then we heard in the previous talk, in many of the previous talks actually, that this is not happening in a vacuum, and so innate cells really matter. And so we wanted to understand more about the signals that are specifically driven by these two vaccine components in the dendritic cells, which are very important for priming TFH cells. And we also wanted to learn more about the capacity of dendritic cells to internalize and express the mRNA, and where does the process occur. And I should say that this was again a collaboration with Norbert, and the study was led by two very talented people in my lab, former PhD candidate Emily Bettini, and a staff scientist in the lab Diana Castano. And you can find most of these data that I'm going to present today in bioarchives, and recently this study was accepted itself, and so hopefully will be out in a few weeks. All right, starting with the first question, is the nucleoside modified RNA immunosilent, or is it immunostimulatory? And really we have to go back to the fundamental discovery of Kati Kariko and Drew Weissman a few years back, because everybody knows that unmodified RNA is highly immunostimulatory, can recognize by different pattern recognition receptor and drive potent production of pro-inflammatory cytokines. And this made essentially the mRNA unusable for vaccination. And so Kati and Drew found that if you replace uridine with 1-methylpseudouridine, essentially you abolish the capacity in vitro of dendritic cells to recognize this RNA in response to produce pro-inflammatory cytokines. And something very similar happens in vivo. And so this is great, was awarded the Nobel Prize and enabled the usage of RNA for vaccination. However, also led to the impression that this nucleoside modified RNA is essentially immunosilent. But we realized when we published this paper in 2021 that this is not true, because if you take mice that lack MyD88, which is an important adapter for downstream TLR, and immunize these mice with a custom-made mRNA vaccine, we found a profound decrease in TFH cells. Whereas when these mice were immunized with a protein plus MTLMPs, the responses were largely normal. So this indicates that there is a sensing of the nucleoside modified RNA, which is instrumental for the induction of TFH responses. And we recently found that the same is true when you use Spikevax, the vaccine from Moderna. So also in this case, in MyD88 knockout mice, we see this drastic reduction in TFH, GCB cells, and spike-specific GCB cells. All right, so at this point, we had this very rudimental model where we know that the two components are both immunostimulatory. So in one side, you have the lipid nanoparticle that drive IL-6, the receptor is not known. On the other end, you have the mRNA that is sensed via a mechanism that requires MyD88. And then through the production of this of an unknown mediator, they drive TFH. And so we wanted to know more about what is this unknown mediator. And because if you would have like an unmodified RNA, this would signal through TLR, MyD88, and would drive type 1 interferons. And since we know from studies in humans, but also from the beautiful paper that Bali published a few years ago, that there is indeed type 1 interferon produced after vaccination with the Pfizer vaccine. So we wonder whether these type 1 interferon were relevant to germinal center responses. So we started our experiment with our custom mRNA vaccine made by Norbert. So in this case, we use hemagglutinin HA encoding mRNA LNP, and we administer before the vaccination an antibody blocking type 1 interferon receptor, so this IFNAR blocking antibody. And then we look at TFH a week after the immunization. And as you can see, we found that while the frequency of TFH and GCB cells was normal, the absolute number was significantly decreased. So meaning that there is a role for type 1 interferon to amplify germinal center responses. But what about the licensed vaccine? Is this relevant at all? And so we did the same experiment using, again, Spikevax. And the result is even more profound, because as you can see in this case, we have a very dramatic reduction of TFH and germinal center responses when we block IFNAR. So there is production of type 1 interferon, which I won't show you, but it's in the paper. So we were able to quantify. It peaks roughly 8 hours post-immunization, and it's mostly interferon alpha, and it does matter for the formation of germinal center responses. So next we ask, what is the target of this type 1 interferon? And so an hypothesis that we had was that this type 1 interferon act not on T cells, but on dendritic cells, because in this case, in certain vaccination or certain infection, it was shown to be key for the amplification of TFH responses. And so to test this hypothesis, we obtained mice lacking IFNAR only in CD11C expressing cells, which for the most part is dendritic cells. And so we immunized these mice either with, so we went back to our custom mRNA vaccine, immunized with either an HA encoding mRNA LNP or with a recombinant HA protein mixed with empty lipid nanoparticle to try distinguishing the importance of the sensing of the two components. And so starting with the HA mRNA LNP vaccine, so when only disease lack IFNAR, we found a phenotype very similar to what I show you with the blocking antibody in our custom vaccine. So there was a significant reduction in the absolute number of TFH and GCB cells, whereas the frequencies were normal. However, when we immunized with the recombinant HA mixed with empty LNPs, germinal center responses were intact. So no alteration was to be found. So clearly showing that there is an impact of type 1 interferon on dendritic cells, which are important for amplifying TFH responses in response to the mRNA component, not much the LNP. Mechanistically, we asked whether these involved the capacity of type 1 interferon to drive the maturation of the dendritic cells that you can track by looking at the expression of the CD80 and CD86 molecules. And in this experiment, we injected the mRNA LNPs, and then we look at disease in the draining lymph node 24 hours later. And when disease lacked IFNAR, we found indeed a profound decrease in the maturation of these cells. And I will show you this data for sake of time, but we also show that when we block IFNAR in dendritic cells in the setting of mRNA vaccination, there is a decreased antigen presentation to CD40 cells. So now, the elephant in the room is really what is the sensor that recognizes this nucleoside modified RNA and then drives in turn the production of this type 1 interferon that are relevant for germinal centers. And the obvious candidate was TLR7. And we thought maybe Catherine and Drew missed that because the ELISA kit was not very sensitive. So let's try it again with the ultra-sensitive ELISA kit or with other approaches. And basically what we did was to use a dual approach. And so we use human TLR7 reporter cell line to track essentially IRF luciferase. And what you see here, our positive control worked very nicely to induce luciferase, whereas different doses of the Spikevax did not. So there was no in vitro activation of TLR7. Furthermore, we also obtained TLR7 deficient mice to be sure we didn't miss on anything, and we immunized them with Spikevax and look at TFH responses, and you can see we found that these were completely normal. So it doesn't really look like TLR7 plays a role here. And so I will spare you the pain to go through 20 slides of negative data, but in essence, I can tell you that we tested with this dual approach, so the cell lines plus the knockout in vivo, all the TLR listed in this cartoon, plus we tested additional sensor, including MDA5, RIGI, STING, and none of them had a clear connection to sensing any of the component of the mRNA LNP in vitro. And some of these are, receptor that would potentially also recognize the lipid nanoparticle or a double-stranded RNA contaminant or DNA contaminant. None of them seem to be essentially giving any positive data. And so the search for these receptors still continues. But what we thought at the point was that maybe we got the interpretation of the MyD88 data wrong. And so maybe MyD88 is not important because it's amplifying the signal of TLRs, but maybe it's important for allowing the signal of IL-1. Because as you can see here, MyD88 is an important adapter in downstream of many of these receptors that are receptors for cytokines of the IL-1 family. And so in this particular experiment, we block IL-1 receptor, which is the receptor for IL-1 alpha and beta, and also IL-18 directly the cytokine. And what you can see now here is that the blockade of IL-1 receptor led to a significant reduction in TFH and germinal center B cells, explaining at least in part our MyD88 previous phenotype, whereas IL-18 didn't seem to play much of a role. However, this is not connected to the phenotype that I show you on type 1 interferon. And this makes a lot of sense because if you read the literature, actually it's known that type 1 interferon and IL-1 are kind of antagonistic cytokines. And so in our setting, actually when blocking IL-1 receptor, the production of type 1 interferon was even higher. What we know is that IRF3 and 7, which are two transcription factors normally very important for the induction of type 1 interferon, are absolutely necessary because if you have mice lacking these two transcription factors, after you give them Spikevax, there is absolutely no production of type 1 interferon. And in parallel, TFH and germinal center responses are completely abolished. So again, the search for the receptor continues, but we know that downstream of this receptor, there is an important role of IRF3 and 7. So now, I hope I convinced you that the nucleoside-modified RNA is not immunosilent. And so now we wanted to know how we can influence the quality of germinal center and the magnitude of germinal center-derived responses. And so to answer this question, we moved to a slightly different system because when you compare an mRNA encoding, an mRNA that encodes for a certain antigen with a recombinant protein plus MPLMPs, you cannot really normalize for the amount of antigen that you give because you don't know how many micrograms of protein are made from the mRNA in vivo. And so to normalize for the amount of antigen, we immunize mice with the recombinant HA, the same amount, that was mixed either with lipid nanoparticles that were empty or containing an irrelevant mRNA. So the mRNA here does not encode for the antigen of interest, but is basically serving the purpose of being an adjuvant. And so we look first at germinal center responses at day 7, and you can see that we found absolutely no difference in the frequency or the number of TFH cells. GCB cells, if anything, seem to be less when the mice were immunized with the luciferase mRNA. But then, on the other end, the frequency of the antigen-specific GCB cells was slightly higher. So overall, the number of antigen-specific GCB cells was relatively normal. So no major alteration in the number of, in the magnitude of germinal center responses. And what about the quality? So we, to assess the quality of TFH cells, we immunized and then sorted the TFH cells a week post-immunization, and then did an in vitro stimulation with PMA and ionomycin. And we tested IL-4, interferon gamma, and IL-21. And as you can see here, when the mice were immunized with the vaccine containing the luciferase mRNA, TFH were making less IL-4 and more interferon gamma. And so the ratio 4 to gamma was changed. On top of that, there was a significantly higher frequency of TFH cells that could produce IL-21, which is a very important cytokine for TFH because it regulates many aspects of B-cell biology in the germinal center. And so in light of these qualitative differences in TFH cells, we wonder whether the magnitude of the germinal center-derived B-cell responses was different. And so we wanted to test the amount of memory B cells and plasma cells found in this vaccination condition. And so starting with the memory B cells, we look in the spleen, 70 days post intramuscular immunization of the vaccine. And what we found was that indeed the presence of the mRNA was a really nice adjuvant signal because the frequency and the absolute number of these the antigen-specific memory B cells was higher when the mRNA was present in the vaccine. Similar data were found for the antibody-secreting cells. And so we, at the same time point, we look at the antibody-secreting cells in the bone marrow that you can see were significantly higher. And in line with that, we also we found a higher production of HA binding antibodies and neutralizing antibodies when the mice were immunized with this nucleoside-modified RNA. So overall, showing that this component is not immunosilent, it is sensed, and it can influence the quality of TFH and the magnitude of the overall B-cell responses. So now, this third point, I will not show you the data for a lack of time, but basically we ask what these two components, how do they influence the transcriptome of dendritic cells? And we found basically that it's mostly the lipid nanoparticle component that changes the transcriptome of the dendritic cells. And I will show you in the summary slide the major finding about that. But what I want to show you in the next few minutes is a few data about the last question. So we ask what type of dendritic cells can process mRNA LNPs and where does this processing take place? So for tracking the capacity of dendritic cells to process mRNA LNP, Norbert made for us this construct where the mRNA encodes for a GFP, and it's encapsulated in a lipid nanoparticle labeled with this lipophilic dye DIL. And so in essence, DIL tracks the uptake, whereas GFP is an indication of the translation of the mRNA. And so we couple these with cytometry panel for detection of different type of antigen present in cells. I'm just going to show you dendritic cell data today. And what we found was that to some extent, all dendritic cells are capable of processing mRNA LNP, but some are better than others. And in particular, CDC2, which canonically are the best at priming CD40 cells and inducing TFH cells, were not only the best at 24 hours, but they were also the fastest. So they were the one that in the lymph node could, with the highest speed, internalize LNPs and translate the mRNA. So then we wanted to know, in essence, where does this processing take place? Because it's very common when you attend a vaccine talk to see this model where you have the injection of the vaccine in the muscle, and then dendritic cells infiltrate the muscle, pick up the antigen, and then carry the antigen to the draining lymph node. And so we wanted to know whether this is actually a true model. And so for this experiment, we collaborated with the lab of Gabriel Victor and the Rockefeller, and they did this fun photoactivation experiment for us using the Kiger mice, which normally express green fluorescent protein, and upon photoactivation, this is converted into a red fluorescent protein. And so the idea here was to inject in the muscle this construct, this type 1 encoding mRNA LNP, and then immediately photoactivate the injection site. And then look at the vaccine draining lymph node 24 hours later with the idea that if these type 1.1 expressing disease are red, they were in the muscle when we injected the vaccine. So this experiment actually showed data that were a bit unexpected because the frequencies you can see here of these type 1.1 expressing dendritic cells that were red was incredibly low. Very just a handful of cells really, whereas the majority were RFP negative. And so this is consistent with the fact that due to the size of the lipid nanoparticle, it's extremely likely that they traffic directly to the lymphatics and reach the draining lymph node where they are internalized by dendritic cells there. And to prove that, we did again a photoactivation experiment using the opposite approach. So now we photoactivated the draining lymph node before the immunization. Then we injected the type 1.1 encoding mRNA LNP in the muscle, and then we look at 24 hours later. So now if the LNP traffic to the lymphatic, you will see red cells that are type 1.1 positive in the draining lymph node. And that's exactly what we found. So we found indeed that 60 to 80% of the dendritic cells that were type 1.1 positive were red. So they were already in the draining lymph node before the injection of the vaccine. So suggesting that most of the uptake occurs locally in the draining lymph node. So putting all these pieces together, in essence, what I show you and what my lab is trying to understand more deeply is what is the contribution of these two components to the overall driven germinal center responses. And I show you that the mRNA is not immunosilent. It is detected and it contributes to the production of IL-1 that signaling through MyD88 is important for TFH differentiation. And then also happening through an unknown sensor, it can activate IRF3 and 7 that then in turn leads to the production of type 1 interferon. Those act on dendritic cells and enhance their maturation and antigen presentation. And you end up overall having TFH cells that have a different qualitative profile. So they make more gamma, they make more IL-21, and potentially directly influence this higher magnitude of memory B cells and antibody secreting cells. Then on the other end, you have the sensing of the lipid nanoparticle. And the part I didn't show you with the transcriptome is that the lipid nanoparticle really induce a pro-TFH transcriptome in dendritic cells that favors the localization of these cells at the TB border through the expression of the G protein coupled receptor EBI2, but also favoring the production of 7-alpha-25 hydroxycholesterol, which is the ligand for EBI2 that contributes to bringing the cells to the TB border. And on top of that, it also induces the expression of CD25 and the soluble form of CD25 that was shown by the lab of Jason's sister to be really important because in essence, what they do is that they create a sink for IL-2. And IL-2 is one of the most potent inhibitor of TFH differentiation. So in this way, indirectly, they favor TFH differentiation. All right, so it really takes a village, and there were many people involved in this study, many people from the lab. I already mentioned Emily and Diana, many collaborators at Penn that generally share many knockout mice for the search for the sensor. And then I already mentioned Gabriel, who helped us with the photoactivation study, but also with the lipstick experiment where we look at the transcriptome of the dendritic cells. We obtain for the custom mRNA vaccine, all the LMPs were provided by Acuitas Therapeutics. And then Andrea Reboldi helped us with some experiment on EBI2, and Roberto did transcriptomic analysis that unfortunately didn't have the time to share. I think I'm on time, I'm gonna stop here.
Very nice, Michela. So it's very interesting. And so it's MIT88 dependent, but TLRs, you know, you saved us the pain, you said, of going through each of the TLRs, which really fits with some of the earlier work we had done, right?
Yes.
But it does seem to be dependent on IL-1. So have you looked at the ASC or the NALP3 knockouts? The inflammasome knockouts or the NALP3 knockouts?
No, we didn't look at any of the inflammasome knockout.
Yeah.
So the TLR, you know, I want to be careful with what I say because I think our approach has limitation, right? And so we know that in vitro, at least the cell line that we use didn't sense any, any of those TLR got activated and the knockout mice, and none of them show a phenotype. I wonder whether though there can be in vivo compensatory effects and
or synergy. Maybe there's some sort of synergy between
exactly.
them. Yeah.
And so, of course, an experiment where you knock out all the TLR possible, that we tested is not feasible. And so I guess we don't have an answer on that. But we also wonder if it's a new receptor or a receptor that is, is not very characterized that could sense this nucleoside modified RNA.
Michela, that was beautiful work and just really elegant. Yeah, I was wondering, have you tried unmodified RNA? Because if you're not completely silencing type 1, I'm just wondering to what extent does it silence type 1 interferon if you have unmodified?
Oh, the unmodified RNA, it's known it induces a very, very strong type 1 interferon production.
In vivo?
In vivo, yes.
Oh,
yeah,
okay,
but we didn't use as a comparator, no. We didn't have that.
Okay.
I think, the scale would be very different. So the amount of type 1 interferon produced with an unmodified RNA would be much, much higher.
Okay, all right, thanks.
Yeah, any additional questions? I think everybody is ready for a break. Thank you so much for a great talk.